You're in the prenatal conference. We'll give you a chance to leave. This might be more comforting. I'm not sure. I'm Ellen Phillips. I'm the president of the ALS Association, Greater Philadelphia chapter, and it's always a pleasure to see and welcome you to a conference of lectures today that are going to be intriguing and easy to understand and should hopefully, when you leave, feel uplifted about the extent of research that is going on now, not just here in Philadelphia, but in this country and around the world. And we have two wonderful experts to talk to you today about that research. Many of you are repeat offenders, and we thank you for joining us again, and that means that we did a good job in the years past. Our first speaker, many of you are very familiar with. Dr. Lucie Brujin is the chief scientist of the ALS Association and going on 12 years in that position, and she's still smiling. That's good, that's good. She has an extensive background in neuroscience and specifically in neuromuscular diseases, neurodegenerative diseases. Interestingly, Dr. Brujin is from South Africa, and when she was considering her career choice, she spoke to her father and he advised, she said, you know, I really like to cook, and so maybe I'll go into hotel management. And the next thing she knew, she was taking a degree in home economics. So from there, she moved into the science field, and we're so fortunate that she did. She did all of her undergraduate and PhD work in South Africa and London. Sorry, I left all my notes at home. I'm gonna get this right. And from there, her experience in neurodegenerative diseases was with Don Cleveland, who was a renowned researcher, and his lab, and also at Johns Hopkins. And her field was Alzheimer's, and worked on mice and got interested in ALS. I hope this is close. The thing about Dr. Brujin there's many things that are impressive, but from the lab, she went to Bristol-Myers Squibb. And having that advantage of being able to be both in the lab and in industry makes your experience quite valuable. And another wonderful thing about Dr. Brujin is that she is able to present her work and the body of work that she has assembled for the organization in a way, in lay terms. Dr. Brujin is known not just by the ALS Association. She has accomplished so much in our field since she turned her attention full-time to ALS when she joined the organization and even before. But she heads up many of you know that the Department of Defense allocates money to the ALS cause, and Dr. Brujin is the chair of an integration panel, ALS integration panel from the Department of Defense, and also chairs so many other important areas, from attending things with the Institute of Medicine, where she works on a program, but there are many other chair positions, including she worked with NIH, NINDS. She has a great relationship with the director of NINDS. I could go on, but she speaks for herself. So I'd like to, at this point, introduce Dr. Brujin, who will give an overview of ALS. And again, those of you that were here last year will see that we've made some very good strides in the past year. Thank you.
Thank you so much, Ellen, for the wonderful introduction. And I too am glad that I've made the switch from home economics, and I'm afraid I didn't do quite as much cooking as I thought I would do in my career and my life. So I am going to try and take you through what has become a very complicated and complex story, but I'm very, very encouraged. I think as we learn more about the science, the potentials of us finding the cause and finding treatments for the disease is ever so much closer. So I really want to start out by thanking all of you and thanking the Greater Philadelphia chapter. I don't think I need to tell you all, but the commitment that this chapter has to research is tremendous. And I list here only a few of the things that they have funded in the last 6 months or so. And so many of them are very recent awards. But we're just so encouraged and so grateful for the support. On the top of the list is a recent commitment. In fact, the commitment from the chapter was not so recent, but getting it all finalized took some time. And I am going to talk about this in my presentation. It's a bit of a mouthful, the acronyms, the NINDS IPS Consortium, so I'll leave that to later. But it's a significant commitment from the chapter, and we're very, very grateful because it puts us as a key player in this consortium moving forward. We had a tremendous drug discovery workshop in March, which was so encouraging to see the industry really interested in the disease and the opportunity to potentially get large pharmaceutical companies like Bristol Myers Squibb and Pfizer to think about developing treatments for ALS. And actually our major contribution came from the chapter here. Virginia Lee, who I just will plug and remind you at the end, funded by the chapter, will be speaking next Monday, so please do tune in. And then the antisense trial, which I will allude to again, an absolutely amazing story of how from lab work we were able to bring, with support from the association and our partners in funding, a trial into the clinic for SOD1. The development of model systems for screening, the Discovery Awards, which are so critical, and these awards are given often to young scientists or novel ideas that are seed funds that enable us to really go into a new area or bring a new scientist on board to commit their time to ALS. Alfred Goldberg, renowned scientist in the field who works on protein misfolding, and for some of you who might have met at a previous symposium, Piera Passanelli, a young scientist who has really built her career and had the opportunity at the Thomas Jefferson Institute to really spearhead her work in ALS with the support from the Greater Philadelphia chapter. So this is just an example, but the dollars, however small, impact, and actually the Philadelphia chapter is of course one of our major contributors to our research program. So I'm going to, it is a complicated tale, and I hope to do it in the most simple terms, and obviously all of you know very well what the disease is, either living with it or being with family that have the disease. But I think it's important for us to understand and look at the biology, especially as we're going to lead into work from Dr. Trainer, to really understand what all these findings mean. And in fact, the challenge of ALS is because of its complexity. If we think of Alzheimer's disease, certainly not a simple disease either, and certainly no treatments and a cure that are effective. But there it's more a focal area. And here, all I'm really illustrating to you is the complex network going on. So from the brain, the connections of a very unique nerve cell in the brain called the motor neuron, which has a tiny cell body and a very, very long projection to where it has to go, from the brain to the spinal cord. And then not only that, but it has to then connect to what its function is to stimulate and activate muscles. So in all of that complexity, we have to think of many, many factors. Not only the regions that are affected, but how they are interconnected, and of course, importantly, the muscles that ultimately waste and cause the complications in the disease. And then I'd like to look into the very depth of what this is all about, and we look at the motor neuron. And if we look here, if I get my pointer right, oh, I did get good instructions, but oh, here we go. This is a motor neuron, and this is its cell body with a nucleus, and it looks fairly healthy. This is from tissue from a normal person, and this is then looking at the disease. And here we see these are projections coming out of the cell body which are abnormally accumulating things. We see here a very strange ballooning. We see here these abnormal aggregates. The system is not working well, and the cells are reacting to it. But it's more complicated than that even, in that I'll show you a neuron with its cell body, and many people draw a motor neuron with one projection, which is correct, that's the axon. But it also has what's called dendrites. This happens to be a mouse motor neuron, the cell body, and all these projections that all have to find the correct connection somewhere. And then the supporting cells, which we now know as also to be culprits in the disease, these are called the astrocytes. And I have just a brief chat about what astrocytes do before the session. In fact, they're thought to mop up excess of glutamate from the area surrounding. Glutamate's a very important transmitter, we call it, that is involved in bringing messages along these long systems. But if too much of it is around, we have to have protective methods to get rid of it. And in part, some of the function of these surrounding cells, the astrocytes, is to mop that up. So they too are engaged in this very complicated process. And in this image I love to show because it really is a beautiful one. It's in fact a motor neuron that at the end branches and connects with the muscle. And this is the dark area, is the muscle. And these are all the connecting points that it's making with the muscle. In this case, we actually have an example of a normal connection where these branches start to disappear or retract or make abnormal connections. You see a very different image. So there's a lot more to think about than just that motor neuron. And then there's a lot more to think about than the simple cause. You know, many people say it was, you know, why have we cracked AIDS? Is it because of all the money that's gone into it? Well, in part that has helped, and I think that does make research move faster. But the reality is the cause of the disease of AIDS is known. Although it's complicated in how it affects and how it needs to find treatments, it's a known cause. In ALS, we have multiple causes. We'll hear a lot more about genetics today, genes. The environment certainly has an impact. Exactly how, we're still unsure, and the likelihood is it's the combination of the genetics and the environment that's critical. And in large part, aging is also a clear risk factor in the disease, with the majority of the disease being in later stages in life. So briefly to talk about programs that we do at the ALS Association, and I draw this schematically. It's kind of the pipeline that they like to draw in industry programs, and actually I have to update my slide because now we don't have a straight line, we tend to make it a circle, because in fact nothing goes in one direction only. It all comes back again, reiterating to try and find out how we can improve things when therapies actually fail. So what this is just really simply trying to tell you is that we have programs that cover all of these areas. First, we help investigators identify what the potential causes might be, and these causes provide us with targets. Targets are just a term in saying something which we can direct our therapy idea towards. And then we have to validate that that target is truly important in the disease, and a prime example, of course, is SOD1, a genetic change we know that is linked to 2% of ALS or 20% of familial ALS. And then once we have that target, there are a variety of things we need to do to try and develop treatments. Model it in a disease model so that we can test the therapies and then bring it through all the way. And although I won't belabor the point, what is very important and a great example for us is after identifying the SOD mutations, superoxide dismutase, which normally is in all our cells to mop up excessive toxins in the cell, they were identified to be linked to ALS in 1993. It is a long time ago, and somewhat frustrating that it isn't simple to treat if we know the target. But we have been able to, through our programs and through support of brilliant academic labs and the company ISIS, we have used this knowledge and taken it through all those steps, and it now is in clinical trials. And some of you are familiar with this, and I'm certainly happy to talk more about it near the end. Critical, though, to a successful program and to a vibrant community of scientists is the provision of tools. We need things like animal models, we need antibodies to track things, those images you saw of the muscle and the neuron, etc. We have to be able to label them. How do we detect abnormal SOD in cells? We have to have antibodies. So building tools is critical. And an area that I'll touch upon again is so important, and that's biomarkers. Clues for us better clues than just thinking about survival, that we can figure out if a treatment is really having an effect. A biomarker could be in the blood, it could be in the fluid that surrounds the motor neurons, which we call the cerebral spinal fluid. It could be an imaging technique where we can look at brain changes. It could be in the muscles to look at muscle changes. These are critical for us to be able to see if indeed something is having a positive effect. And it's partly why now the trials are so challenging, because our best marker of an effect is the change in survival. So we have to have very long trials to get to that kind of an endpoint. Certainly another challenge that many of you in this room are very familiar with and can be frustrated by is the slowness of diagnosis and the need to diagnose the disease earlier. This again is very, very important, not only for you as families to know how to prepare yourself, but more importantly, I think, to be able to start treatments much, much earlier, because sparing as much of the damage that is already done and starting early will ensure that when we do find treatments that are significant, we can actually slow or even halt the progression and not have come to a point where things have declined so far. So what do we fund in our programs? We focus a lot on genetics, and you'll hear about that today, and I know that Dr. Traynor will highlight why that's so important for us. We focus on initiatives that are consortia, and stem cells are of particular interest. As I said, building model systems for the community is critical, and at Jackson's laboratory, who really is the provider for most of the mouse models in this disease, we have now built a partnership that is encouraging scientists who build new models to get them centralized as quickly as possible, even prior to publication, so that scientists worldwide can have access and we can do studies more quickly. Biomarkers I did tell you about, clinical trials are absolutely critical. And so why are our research programs so important to us? Well, we have an ability, NINDS is clearly the NIH of the government, provides most of the funding for ALS research. But what is critical is that we are able to fund at any time of year, all over the world, and pick novel ideas. We are flexible in that. We are able to build infrastructure type grants, and I'll talk about clinical trials. We seed opportunities, so many scientists have said to us how important this small grant that we gave them was, because now they could get government funding. Sometimes there are risky ideas that are not sure to succeed, and after an initial funding, we're able to ensure that they then succeed with that data to get funding from the government. What I think is truly important, and I value the time that I had at Bristol Myers Squibb to recognize these opportunities, is building partnerships with the industry. The academia is critical in developing and understanding the biology of the disease, but ultimately the treatments will come out of the pharmaceutical industry. And so opportunities to spearhead these things. In many cases, we actually fund small biotechs because it might seem counterintuitive, but as they have not focused on ALS and because the risk in succeeding is quite high, we have helped to seed funding so that they then will be more confident in taking it on board. And I bring back the story of ISIS because it is the perfect example of this. A biotech company in San Diego had never worked on neurological disorders, had never used their technology, which is very novel, for a neurological disorder. They worked together with Don Cleveland and Richard Smith and other academic colleagues to try and explore what was then thought to be a quite crazy idea of taking a system to stop the production of something we don't want, and in this case it's SOD1. And so they provided the reagents, we provided the funding to Don Cleveland, and actually, lo and behold, this technology worked. And so then, once it's worked in the mouse model, we had to fund the very expensive studies that the FDA requires, and we did that. We committed the funds to that, and then took it towards being in clinical trial. Now ISIS actually supports work in Huntington's disease, in SMA, and in myotonic dystrophy. All diseases that before were not considered a possible target for this kind of therapy. So it's critical, and in fact now they don't need our funding. They're very well supported, and they are preparing to take the next steps, and you'll learn more about what that is when we learn about the chromosome 9 gene. And finally, I think the most important thing that our research programs do is bring scientists worldwide together to discuss data. I'm often asked, well, how do we know if we're not overlapping our funding? How do we know if people have discovered something and they haven't shared it, we start to fund it again? And I think it is because the community, although very large now, is always interconnected. There are meetings, there are conferences, there are discussions. As Dr. Trainer will show you, we don't do this alone anymore. It's teams of people that come together to figure it out. And so we help facilitate that by having workshops and symposium to bring them together. And in particular, I think stimulating young investigators through our fellowship program. We have a clinical scientist award because what's also critical, and we've learned over the years as we've become much more involved in drug discovery and developing treatments, that we all have to work as a team together from the early basic science to the clinical aspects, because it's really understanding the whole spectrum of the disease that you can improve the kinds of studies that you design. We have several pilot studies and clinical trials now. Clinical trials, to put a perspective on it, can cost anywhere in the range of 25 million. We were talking a little bit about glutamate being a challenge for the disease and the lack of being able to remove it. Ceftriaxone is a trial at the moment being done to see if we could improve the removal of glutamate, and that trial in itself is 25 million to run the trial. So it gives a perspective on it. So no, we don't spend 25 million on pilot studies, but we spend anywhere from 300,000 to 500,000 to help improve the study by either finding out what dose should be used or improving the biomarker for the study. Or in the case of refining which are the people we should have in the study, we currently have an SOD1 trial looking at arimoclomol, because maybe, and in fact I think it probably is going to be the case, because ALS looks so different in many patients, maybe there are some that will respond to one thing but not another, so we're going to have to segment out the population. We are now also launching on a new program. We have funded in the past in a small way, but we intend to invest more robustly in this program to actually improve the living with the disease for patients with ALS, and that's what our management grants are. And one example, we're just in the planning process about to fund a test to see whether the diaphragm pacing, some of you have heard of this technology. It's a way to keep the diaphragm from moving and so help respiration, and it's already been approved as a device. But we are not absolutely sure that this is necessarily better than other things that are available to help. And in fact, there are some claims that this improves survival, but insufficient data supports that. So before just suggesting to everyone to have this approach and encourage this kind of technique, we felt it critical to do an improved and better clinical trial which will test that, and that's the goal of this kind of a program. And then finally, we have supported a clinical trials network, and this has significantly helped us much more quickly get clinical trials into place. So this is something you'll see again today from Dr. Trainer in a different perspective, but this is what I think makes this field so incredibly exciting. I talked about 1993, and actually that coincided with the time that I started to work on ALS, and this was what was known about the cause of familial ALS, that there were mutations in superoxide dismutase. And years went by until 2006, 2007, when we found the next two mutations, TDP43 and FUS. And not more than 6 months ago, this mutation was found. And just to highlight the significance of this finding, such a big proportion of familial and sporadic ALS. And so we haven't got a long way to go. We do still have some way to go to find the remaining genes. But what does it mean to us that we have all of this information? Because ultimately what we want is treatments. And this is just showing you the structure of SOD1, superoxide dismutase, the fact that all these colorful things here are changes in the coding, in the message on this gene, which results in something abnormal in patients, in abnormal superoxide dismutase. We know that probably what is happening is something toxic and new, because actually we have one normal copy and one copy that has this mutation in those that have the familial ALS. and it still functions quite normally to get rid of toxins. So whatever it's doing in the body is something abnormal, and we've spent years and years figuring this out. We all have our theories, but the idea of the SOD1 trial is just to get rid of that protein that is abnormal, because whatever it's doing is bad. We don't need it, at least at reduced levels, and so we remove it. But what else it has done for us, finding this gene, is to develop model systems, all kinds of model systems, from worms, fly, zebrafish is not on here, being able to take these cells from that mouse model and put them in a dish and look at what goes wrong with them, mouse models, rat models, and one might wonder, well, why all these model systems and why so many different ones? And each of them has a different value. What is quite amazing is you can take the human gene, which carries the changes in the that are connected to the ALS patient, manipulate it so that you introduce it into the mouse genetics, and it develops a disease which is remarkably similar to ALS. At midlife in these mouse models, they start to drag their one limb, it spreads to the next and goes to the forelimbs. So we are able to look at these systems well before the disease looks obvious and start asking, what are the pathways that are leading to this? This is critical because we can't do that in humans. We can't see what were the early events that happened. What does that mean for the biology of the disease? And it's things like this that we try and investigate so we have clues of the pathways that are involved and the things that we can do to try and treat in the human disease. The worm is used because actually, amazingly, it's got a very simple nervous system, and you can see it in live time. And here, what we have done is put into the, I'm sorry, I knew I would do this. What we've put into the worm is this abnormal SOD, and we can start to see where it's moving and where it's tracking. And we can then put compounds on these worms and say, are there any compounds that can change the behavior of these worms? When you put the mutant protein in it, this SOD1, they have problems in movement and they don't move so far as well on the dish. When you put chemicals on top of them, you can see which chemicals are effective in changing that movement, and they provide clues for us for the development of treatments. And then this is another model system which is very much used recently and it has been very successful. In fact, you can also in live time, it's see-through, you can see all the pathways that you want to trace. And here, if we look here, this is looking at axonal branching and dynamics. So when we talked about that motor neuron with a long projection and where it's going, there seems to be some abnormal changes when you put the mutant protein into the worm. And then you can start to ask, again, which treatments or which genetic changes happen to resurrect that abnormality. And some wonderful work has been done that has given us strong clues about new pathways in ALS. But with all this brilliance and with all this exciting research, of course we have learned that the disease mechanism is a very complicated picture. And all you need to take away from this image is, here is your schematic motor neuron, which is clearly not quite what it looks like. But what is important is it's the muscle that is involved, the surrounding cells that are critical, how one gets things from one end of the motor neuron to the muscle is also very important. We call that axonal transport, and a very complicated system that can probably go awry when things are wrong in the cell. So these are all clues for us. They're all the targets we have to start developing therapies. And without this picture, we would not know exactly where to start. We still are challenged what is most important, what starts first, but it gives us tools in to start testing all these ideas. And actually, what's probably going to happen is there isn't one main method that these cells are dying. There are multiple methods. And so what we have to do is use multiple therapies to treat the disease. It's already starting to happen. Of course, we have Riluzole, very modest effect. We now have a new drug in trial, dexpramipexole, together with Riluzole. We imagine that what will happen is that each of these will have a small effect, and put together, because they're working on different pathways, will likely increase and enhance the effect. So we do believe that it's going to be multiple approaches that will be important. So I only briefly touch on this, and I think I really do encourage you to listen to Dr. Virginia Lee on Monday at the webinar. She has been a key in many of these studies, but it is to remind me to tell you that we, for very long, were focused on a very motor neuron-centric disease. And yes, it is the motor neurons that die, but actually in some cases, in quite a few cases of ALS, there is also a dementia component, which we call frontal temporal dementia. And it is interesting, as we lead into Dr. Trainer's presentation, that the gene that he has found is linked both to ALS and frontal temporal dementia. And all this is showing you is that there are many different genes involved, FUS, TDP43, that are involved both in frontal temporal dementia and in ALS. And so what is intriguing, and I think what we need to start dissecting, is why is it that one particular gene can either cause a disease that looks more like a motor neuron disorder with the function of the motor neuron dying, or one that has dementia, or one that has both. And so actually, we represent ALS as a spectrum, from on the very left, a very pure ALS-like phenotype with motor neuron involvement, and on the very right, a very pure clinical phenotype of a frontal temporal dementia, and then anywhere in between. It does make it very difficult to think of treatments, but I think it's very important that we know this, and so that we can carefully dissect out what kind of treatments, what kind of populations are going to be sensitive to which different approaches. And then briefly to talk about stem cells, I often in the audience hear the comment, well, stem cells is going to be the cure, this is all it. I don't believe that that is the case. I think that they're extremely important, and I think I'm very excited at the progress the field has made, but I think we have to have a balanced view on this. First of all, it's extremely difficult to inject these stem cells, get them to the right place. If I remind you again, the difficulty and the architecture of the disease, how widespread it is, where do we put these cells, do they truly reconnect? Many, many questions. But stem cells have provided us with many other opportunities. This is just to highlight, and I can certainly in the question session address the stem cell trial. This is just to remind me to tell you that there is a stem cell trial in the US, NeuralStem, and it's a very, very complicated trial in that you have to inject the cells into the spinal cord, and that's done with this apparatus, this may not look sophisticated, but it took a lot of work to put that together so that the cells are injected in exactly the right place, and that the apparatus is able to move and shift with the breathing and the movement of the spinal cord such that damage isn't done to areas that you don't want to damage. But it's a very invasive way of putting these cells into the right place. And we don't know yet from the trial that this works. We know at least that it's safe. But what stem cells can also do is build model system for us. And this is what IPS is, induced pluripotent stem cells. And this is a tremendous technology from just the last few years. There's always been a debate about the embryonic stem cells, the concerns ethically. This actually circumvents all of that, and what you can do is take skin cells from a person with ALS, you manipulate it with a few genes, and you can reprogram those cells to become embryonic. You can then shift those cells to become motor neurons and astrocytes, the support cells that we talked about earlier. And then you can start to take them from people that have the disease but you don't know anything about their genetics. You can take them from those that have the TDP43 mutation or the chromosome 9 mutation. And you can look at them in a dish and ask, how are they different from those that don't have the disease? And in fact, one might think, well, that's an impossible task, and will you see anything? But indeed, you do. If you take cells from someone who carries the genetics of ALS, whether you know the genes or not, and those that don't have the genes survive longer in a cell culture under certain paradigms than those that do. So we do know that there is a change. And in fact, fabulous technology, and really this is a microscope with an automated plate, enables us, and this is an investigator, Dr. Steve Finkbeiner at Gladstone Institute. He looks at these cells from these patients in live time and looks over time to see what is happening to them. And then you can manipulate the system. And he captures that. This is really a rather complex thing to say that this is all part of it. You collect a lot of information and data. You've got to find a way to interact and get that data to mean something. And ultimately, he's taking images over time. And he is, with the system, being able to trace these abnormal clumping that happen in the cells that we know is a problem. We see that in humans at end stage. And he's now manipulating with certain drugs to see if that can be affected in a positive way. And so what is the NINDS, the National Institutes of Neurological Disorders and Stroke, IPS Consortium? Well, we were very fortunate several years ago as part of the Recovery Act and the Obama stimulus funds to get money through the NIH for an ALS consortium. And these are the talented investigators that are part of that consortium, either providing the patients with the material. Kevin Agin comes from a background of stem cell technology, very talented. Hyperion is the company that's working with us and doing this. And we have, through this consortium and through funding initially from these stimulus funds, been able to build banks of cells that are now well characterized from people that carry the SOD mutation, the TDP43 mutation, and really provide them ultimately to scientists worldwide to do various experiments. And because the stimulus funds ran out and there wasn't a renewal, and the value of this consortium is so important, we at the ALS Association felt we needed to continue this in partnership with NIH and now in partnership with many, many companies, because in fact the companies see this as a wonderful opportunity within their company to use human cells. Remember, in fact, the time that I was at Bristol Myers Squibb, we used rat and mouse cells to study aspects and look for drugs. But we've actually learned that there are differences between the human cells and the mouse cells. You probably say, well, I knew that. But in fact, they do react differently. And it's very important that we now have cells that come from the person that has the disease and the human cells. And this might lead us to better clues of treatments that might work. And this is just to list all these cells that are now housed centrally for people to use worldwide. Oh, and this does not image very well, but this is just to highlight to you that I mentioned to you that we have a centralized repository for animal models as well at Jackson Laboratories. And what is fascinating about working with these mouse models is they don't always react in a predictable way. This is a new mouse model that was developed for the TDP43 mutation, and this is showing you a survival curve of those models. And what's very important to note is that actually the females survive a very long time and the males do not. We don't know the reason for that yet, but it's very important to know that, because imagine if now scientists are putting together a trial, a drug trial in the mouse model, and are only using females, or are using a mix actually of females and males. And they say, gosh, there's a real extension here. It must be the drug that's doing that. Well, actually, in this case, it's only the gender that changes it. So this is a repository that actually houses the data centrally and characterizes these models very, very carefully so that we provide the right information to the investigators and we can ship these models out. So it's an example of how we can really help facilitate researchers to do things much faster. And actually from these mouse models you can also develop stem cells and generate these motor neurons. And this is valuable as well to develop model systems. And this is just to show you here that when you do that from a mouse that has this particular mutation, the TDP43, you get some very abnormal looking cells which you can now test in a system to see if you can improve them. So my final few comments are looking at biomarkers, and I think what is very exciting about this is an important molecule, and I won't give much detail about it other than that it's very prominent in motor neurons and very much in that axon, that long process. And all you need to realize from that is that when we take blood and we measure the levels of this protein, in control samples it's very low. In ALS patients it's very high. And this team are now trying to look very carefully, and here this is looking even over time. What it looks like over time is always much higher than in control patients. Is this going to be a valuable diagnostic? And can we use it in clinical trials to show if there are changes in the levels of this protein? Have we then got a compound that is effective? So again, it was trying to tell you that there might be clues in blood samples or in the fluid around the neurons that can lead us to developing easier tests for diagnosis and for drug trials. This is a study that we have funded in Boston, and Sue has devised a technique which you can much more effectively measure muscle changes. And clearly that's actually very important because we know that it's the decline in the muscles that is ultimately what happens in the disease. And in fact, this is now going to be put into the clinical trial at Biogen, the Empower study, in its next phase to see if they can more accurately assess whether the drug is working. And I'm not going to tell you the biology behind it's a little complicated. This is to show you that this is the clinical trials network that we support throughout the US. And what do we mean by that? We provide funds to help build training, infrastructure, centralization of data. When there are things like the device used to test the muscle measures, we make sure it's standardized in every site, because what we have to do for clinical trials, because of the numbers of patients in each region and the numbers we need in trials, is we have to combine all these different centers. So we might have 20 from one site, 30 from another site. And the danger of doing that, if things are not standardized and accurate, is that you might not get the same outcomes in each of the different centers. And this assures that the quality and the standard and the data coming from it is very, very clearly defined and accurate. And I would say that because of this financial support and because of the changes in the field, there's been a tremendous increase in the number of very, very well-run clinical trials in the US. And these are the trials, and I'm happy to spend more time on them, but I know that there might be very specific questions, so I won't go through every one of them in detail. Empower is the dexpramipexole trial at Biogen. Very briefly, it's completely enrolled. For those of you who were at advocacy, they saw Dr. Doug Kerr speak. They know that they hope to conclude the trial at the end of this year. It does take time to analyze the data, but the hope is that next year they will know the outcomes of this trial. The goal of this, we think that the product is working, or the drug is working at the level of the mitochondria. I didn't talk about those, but they are important organelles that are the powerhouse, the energy in a cell, and they are very important in the motor neuron. And if that is dying, the energy within the cell is depleted, so it might well be working like that. The CK compound, the complicated number, is from cytokinetics. It's a small trial that has just been completed. It's a very interesting compound that works on muscle contraction and improving muscle. And I believe that we hope it was, the early data looks interesting. If it is effective, it will be very important in combination with other treatments so that the quality of life is maintained at its best as we get newer treatments. So it will be very important as an additive. The neural stem trial we've talked a little bit about, and the antisense trial. This is not the complete list, but these are the major ones that are currently ongoing. And I think with that, I will close and ask for questions. And again, remind you that on Monday, Dr. Virginia Lee will be speaking. And I think the value of these webinars, I always hope that I pick someone who might be able to explain a very complicated field, but it is an opportunity for you to ask questions, to learn about the science directly from the scientists, and it's always been extremely well attended, so we hope to have you on the phone on Monday. And with that, thank you very much, and very happy to answer any questions that you have.
Hi, Lucie. A really basic question. What do we mean by the term translation?
So translational means a lot of different things, and if you were to look it up even at NIH, it's different things. Generally speaking, in terms of translational, that I use it and that many biologists use it, it's the idea that you translate an idea from the laboratory to the clinic. But it could also be translating at a much later stage to the clinic. But the bottom line is that you are taking an idea and you are taking it all the way into a treatment. That's translational. So any research that is, so we argue that the DoD ALSRP program, and I know that that's probably where it comes from, is translational because we are testing drug compounds, we are testing ideas with the very specific goal of developing a treatment for the disease. And it's very different to where we say we fund the basic research. When a scientist comes with a proposal to test how the cell interacts with another cell, they don't have translation in mind at all. They want to understand that mechanism. It feeds into it later, but that's not translation. It has to be closely linked to a trajectory that will take it to the clinic. I know it's a lot of information, and it's always hard to know what to select to present, but
There's one over here.
yes.
Has the ALS registry been of any help in any of these trials or any of the studies?
It's early days, so unfortunately it's hard for people who want an immediate registry right today. It takes time to populate it, and it's only as good as how many people get into it. So it's a plug. Please, please, please be involved, because the more people and patients we have information on, the more valuable it is. And what just came out live on Friday is a tool for scientists and clinicians to go into the registry and let people know that there are clinical trials available, and the CDC that can then maybe even select, they can send an email out to all the people in the registry, do you want to be involved in this trial? Or they can provide information to the scientist as to which criteria, if they are given criteria, which criteria match that trial, and then they can trawl down and give the information. So that's the beginnings of, for example, valuable use of the registry.
Can they only be in one clinical trial at a time?
That depends a lot on the trial. Generally speaking, yes, although I know for the dexpramipexole trial, you can be on riluzole and dex. There might be some unusual situations that you could be on 2 trials, but in reality that would be very complicated, because you wouldn't know which trial was having the positive effect. If you're on dex and you're on whatever it is, you won't know then which is the one that is improving your disease. So in general, the reality will be that there's only one trial agent being used. Yes.
Lucie, when we learn of a cluster situation and it's reported, is there anything, how does that work in terms of whether it's examined?
So a variety of things. I mean, I think there's often a sense that there are clusters in many areas. I think the registry is going to be extremely valuable for that particular area of research, because it's very challenging. Before you can really be sure there's a cluster, there's a lot you have to know about that population, the demographics, the movement of people in that area. A very simple example, we worked when I first joined the association, there was believed to be a cluster at the Kelly Air Force Base in San Antonio. And in that particular instance, we worked with the chapter, and we got an epidemiologist and a clinician to form an advisory team, and they actually devised what is called a mortality study in that particular area. And you compare them with other diseases like cancers in that area, with a population-based and a control-based. And in fact, there was not an increase in mortality. That doesn't mean to say for that period of time there wasn't. It doesn't mean to say that over time, with renewed data, it might change. But so it's very difficult, and it needs to be done very carefully together with an epidemiologist and the chapter if it's felt that it's strongly an area of concern. One more question here, Chris.
I've just heard of exome testing, if that's the correct term, which is fairly new, been approved to look at exons. And
I'm going to leave that to my colleague to answer, so ask him again. We have an exome expert. I think that's probably, we probably have time for questions at the end if you have things that come up later. Thank you very much.